Good morning, students. Now we have to study about the anatomy of plants. What is anatomy? That is the internal studying about the internal structures, internal parts of the plants. Okay. Already we saw that internal structures of root and the introduction of anatomy. Now we have to study about the anatomy of stem. What is stem? That is the above the soil. What are the region, what are the parts that is present in the stem, in the plant pot, that is a stem regions. In the stem regions, that is a phototropic. For an example, below the soil the roots are growing towards the soil, penetrating the soil, that is a geotropic, because of that is growing towards the and penetrating the soil. Okay. Above the soil, the stem is growing towards the sunlight, so that it is a phototropic. Okay, stem is a phototropic. Then, what are the parts is present or present in the stem? That is a buds, lateral branches, many flowers, leaves, and all the things. Okay, that are stem regions. Okay. Now we have to study the internal structures of stem. Okay. Then what is dicot and monocot? Cotyledons means what? Cotyledons means that is while the seed is germinating, the covering or protecting leaves, some leaves are present with the seed, or while the seed is germinating, with the seed some leaves are present, that leaves are present cotyledons. Okay. If the seed is having the two cotyledons while germinating, that is a dicotyledons. The picture is also given. A very simple picture is given, and two embryonic leaves are present. That's why that is called the dicot plant. Okay, the cotyledons is shortly called cot. Then monocot plant. If the seed is having only one cotyledon, that is called only one embryonic leaf is only present, that is called monocotyledon or monocotyledonic plant. That is simply called monocot plant. Okay, this is a dicot and monocot plants. The dicot plants and the monocot plants are having stem regions. The internal structures of the stem are only we have to see. Okay, now we have to see some pictures of dicot and monocot. First dicot: sunflower, pea, mango, tomato, grapes. These are some examples pictures, some example pictures of dicot plants. And monocot plants also: banana, coconut, garlic, maize, and pineapple. These are some examples of monocot plant. Okay. Now we saw that dicot and monocot plants introduction. Okay. Now the internal structure. How the internal structures is organized in nature. How the God has created the internal structure of the plants. That thing we have to see. Okay. If we cut the internal structure of the stem, we can see this, in which commonly we can see that round structure. See, that is the internal structure of dicot stem, shortly called TS of dicot stem. Here the example plant is sunflower. Sunflower plant we took. Okay, in which the outer region of epidermal hairs, and the epidermal hairs is attached in the epidermis outer layer. Below that hypodermis. Then the largest space is there, in which many cells are present, that is a cortex. And the innermost round layer is present, that is a endodermis. In the endodermis many vascular bundles are attached, and inner to the vascular bundles some largest center region is present, that is a pith. These are some of the parts are present in the dicot stem. Okay, if we cut a small region, if we cut a very small region, that thing is enlarged and absorbed. This thing. Okay, and now each and everything we'll see. From the epidermis to the pith, we will see individually at first. Epidermis. See, epidermis. Each and every part is cut and we will study individually. First one, epidermis. That is the outermost layer. That is the outermost layer and it is only protecting the internal parts of the plant. Okay. That is a single, that is made up of a single layer of parenchyma cells. The outermost layer that is made up of single layer of parenchyma cells. Then what is a parenchyma? That is a parenchyma cells is a filling cells or that type of cells is having a very thin outer cell wall and having a cellulose chemical in their cell wall, and that is a storing cells also. Now it is serving as a protective in function at the outer region. Then see the picture. A small outer coating is that is also present, that is a cuticle, and some hairs like regions are also present, that is a epidermal hair. Okay, that is the epidermis layer in which the cuticle coating is present. The cuticle coating is serving the protection in function, and the some epidermal hairs is also that is also protecting from some defense, from some affecting also. Okay, then cortex. Inner to the epidermis, cortex regions is present. The cortex is divided into 3 layers, that is a hypodermis or upper cortex, and the middle cortex and the inner cortex. One by one we will see. First one, upper cortex or hypodermis. Just below the epidermis, 3 to 6 layers of collenchyma cells are present. Collenchyma, a type of cells. What is collenchyma? That is a unevenly thickened cell wall containing cells, because of in their cell wall hemicellulose and pectin, that type of chemicals are present in their cell wall. That is a collenchyma cells. That cells are filled up in the hypodermis cells. And what is its function? That is a mechanical support. It is also giving some support to the plants. Okay, that is a hypodermis. And middle cortex, that is a second or middle regions of the cortex, that is a chlorenchyma. In the middle cortex, what are the cells are present? Chlorenchyma cells are present. Chloro means chloroplast. Plastid, you know, red color plastid, green color plastid, and colorless plastid, that is a leucoplast. Red color plastid, rhodoplast. Green color plastid, chloroplast. Colorless plastid, leucoplast. So some type of plastids are present in the plants only. Now green color plastid, that is a chloroplast. That is chloroplast or chlorophyll pigment, that is present more in these chlorenchyma cells, so that it is a chlorenchyma. Okay, chlorophyll is present. Okay, what is its function? 2 to 3 layers of chlorenchyma cells are present. Chlorophyll, by having the chlorophyll, it can do photosynthesis. Photosynthesis means, photo means light, synthesis means creating or producing something. What is created? Food is created. Okay, that food is created so that it is a synthesis in the presence of light, so that food is synthesized, so that synthesis. So photosynthesis means food preparing process is happening in these type of cells, so that it is a chlorenchyma. Then inner cortex, the last regions of the cortex, that is a inner cortex, in which a few layers of parenchyma cells are filled up. Okay, what is parenchyma? The parenchyma cells is a round or oval shaped. The cell wall is made up of cellulose or hemicellulose. Okay, the many intercellular space is present in between the cells. That is a very, very important in nature. Okay, then what is its function? Gas exchange. Unwanted gas is released from the cells and wanted or required gas is uptaken or getting from outside. That is a gas exchange. CO2 gas is released, O2 gas is getting. So that is a gas exchange. Then food storage. Okay, in the chlorenchyma cells, the previous region, food is prepared. Food preparing process is happening we saw. Okay, that food is stored in these parenchyma cells. Okay, not that food is stored in the form of what? For an example, carbohydrate, that is a food that is prepared, that is stored in the form of starch. Okay, not only the starch, some oil content, some tannin, some resin. So this type of food content, this type of food is stored in these parenchymatous cells. Okay, then next one, endodermis. Next to the cortex, endodermis. Okay, endodermis, that is a single layer of barrel shaped cells. Okay, it is having many starch grains. It is having starch grains and looking like a sheath around all the vascular bundles, so that it is here, it is called the starch sheath. Okay, then stele. Stele is also like a cortex. Stele is also divided into three: pericycle, vascular bundle, and pith. Stele. Okay, to the endodermis, cortex is over. The stele is started from the pericycle. Okay, pericycle, see the picture, pericycle here, the blue color, above the blue color some blackish regions is present as sclerenchyma cells. From that is a pericycle. Here stele, sorry, pericycle. The multi-layered parenchyma cells that is present above the vascular bundle or at the top of the vascular bundle, that is altering with some sclerenchymatous, parenchymatous, sclerenchymatous, parenchymatous. The altering patches of parenchyma and sclerenchyma, that is a pericycle. Okay, then vascular bundle, very very important in the plants. Vascular bundle, by observing the vascular bundle also we can see that it is a dicot or monocot, or it is what type of plants it is. That thing we can see or we can find. Okay, vascular bundle, that is a conjoined. Conjoined means what? For an example, in the vascular bundle, the two important things are present: xylem and phloem. Xylem, that is conducting water; phloem, that is conducting food. Okay, in which the xylem, okay, xylem is present towards the inner side. Okay, conjoined means xylem and phloem that is present in the same line, that is called the conjoined. Xylem and phloem that is present in the same line like a jointed one, that is a conjoined. But in radial vascular bundle, xylem is present in one radius, phloem is present in one radius. In a round vascular bundle, inside the vascular bundle, xylem and phloem that are present in different radius, that is called the radius vascular bundle. But here the xylem and phloem are present in the same line, that is a conjoined. The next one, collateral. Phloem lies towards outer side, xylem lies towards the inner side or towards the center, that is a collateral. Then endar. Endo means inside. The protoxylem towards the inner side. What is protoxylem, metaxylem? Protoxylem means first formed xylem and the smallest xylem also. Metaxylem means the old xylem and that is a larger one and having or looking like a vessel like structure or a large tube like structure, and that is conducting water more or bulk amount. Okay, now two type of smaller xylem, protoxylem, larger xylem is metaxylem. Here protoxylem lies towards the inner side means endar, protoxylem lies towards the outer side means exar. Now here endar xylem is only present and open. Open means what? See the blue color cells are phloem and the rounded cells are the xylem. Okay, between the two xylems, Gampium cells is also present. That is Gampium cells, that is 2 to few layers of cells are organized in between the phloem and the xylem. If the phloem cells are present, that is a open vascular bundle. If the Gampium is not present between the phloem and the xylem, that is a closed vascular bundle. Here it is present normally in dicot regions or dicot stem, the Gampium is present. Okay, here all the vascular bundles, see the structure. All the vascular bundles are arranged in a ring, arranged in the or attached in the endodermis, that is looking like a ring like structure. Okay, That is a vascular bundle structure in the dicot stem. Then pith. Next to the vascular bundle, inner to the vascular bundle, yes, in the central parenchymatous tissue. Normal parenchyma already we know. The central parenchymatous tissue that is present next to the vascular bundle, at the center region many parenchymatous cells are present as freely, and that is used for the storage of food content. That is a pith. Okay, in the dicot stem only the pith is present. Okay, then now internal structure of dicot stem is over. Then monocot stem. What is monocot? Already I told. The monocot stem, dicot stem over. Monocot stem we have to see. If we cut the monocot stem, we can see the structure. See, in which also the outer layer is the epidermis, and the below to the epidermis many parenchymatous cells are present, and the ground regions are filled by the many parenchymatous cells. That is a ground tissue. In the ground tissue many vascular bundles are scattered here and there. Okay, so these are the internal structure of monocot stem. Now a small region is enlarged in the monocot stem. See, small region, cuticle, epidermis, hypodermis, and inner ground tissue, and many vascular bundles are here and there. Okay, now here also one by one we will see the outer region. See, epidermis. Epidermis is only cut. That is outermost layer. That is the outermost layer. Single layer, outermost layer. That is also protecting the inner parts of the plant, in which the parenchymatous cells are only making this layer here. Very, very important. Hairs. Epidermal hairs are absent, and stomata that is also very less in number. Okay, epidermal hairs. In root that is present root hairs. In stem that is a epidermal hairs. That is giving some support to the plant stem. Okay, then what is stomata? Stomata is a minute holes or minute openings in the leaves. In the leaves, the minute openings are present in the leaves. Sometimes in the greenish part of the stem, the stomata is also present. Here, very less number of stomata are present. The stomata is doing or serving what? Stomata is a transpiration. Excess amount of water is evaporating in the form of gas. That is a transpiration through the stomata. The transpiration is happening here. Very less number of stomata is present here, so that transpiration is also very less through in the stem regions. But then transpiration is happening only by the leaves only. Okay, then after that epidermis, hypodermis. See, below the outer epidermis, some hypodermis layers or few layers of sclerenchyma, sclerenchyma, black colored sclerenchyma cells are present. That is interrupted by the green colored chlorenchyma cells are also present. Already we saw that sclerenchyma, chlorenchyma, all the things are present. Sclerenchyma means the outer cell wall is made up of lignin content that is present, mainly mechanical support. Wherever the sclerenchyma cells are present, that region is very hard in nature. Okay, for an example, bark and the surface of the seed, that are all very hard in nature because of the sclerenchyma cells are present because of its lignin content of in the cell wall. Okay, then ground tissue. Below the hypodermis, in the ground regions, the mass of the parenchyma cells are present towards the center. Here there is no differentiation. In the dicot stem we saw some differentiation. Cortex, endodermis, pericycle, pith. Like the dicot stem, here there is no differentiation of cortex, endodermis, pericycle, pith. Here there is no differentiation. So all the ground tissue is made up of parenchyma cells only. Then vascular bundle. Here the vascular bundle, see, skull-shaped structure. Skull-shaped structure, 2 eyes, 1 nose, and a large mouth-like structure. That is a skull-shaped structure. Okay, that is a skull-shaped structure. Then conjoint. Already conjoint. What is conjoint? The xylem and phloem that is present in the same radius, and collateral. phloem lies towards the outer side, xylem lies towards the inner side, and the end or protoxylem lies towards the inner side, and the closed. Here closed very very important. Remaining things are present. Remaining things are like dichotom. Only the closed. That point is only a significant point of the monocot stem. Closed means between the blue color phloem and the rounded part, xylems, between the two phloem and the xylem, between the two things there is no cambium. Then sclerenchymatous bundle sheath. Okay, surrounding the vascular bundle, some sclerenchymatous cells are present. So that is a safety layers of the vascular bundle, so that it is looking like a sheath also. So that is a sclerenchymatous bundle sheath. Okay, then next one, xylem. The xylem vessels, here two metaxylem and one protoxylem is present. Smallest xylem protoxylem and the largest xylem is the metaxylem. Here two metaxylem, one protoxylem is present in the xylem, that is looking like a V or Y shaped. Then protoxylem, the smallest protoxylem that disintegrates and form a cavity like structure. So that is that only forms a skulls, that only forms a mouth of a skull. So overall things, by looking the overall vascular bundle, we are looking as a skull shaped structure. Okay, then phloem, sieve tube and companion cells. Here not a type, only tube. Sieve tube and companion cells are present. That is a conducting part of the food. And phloem parenchyma, phloem fibers, FIBRES, phloem fibers are absent. Phloem parenchyma and phloem fibers are absent. That is the constituents and or components of the phloem cells. Then pith, here there is no pith, because of that at the central part is present freely, so that many parenchymatous cells are filled or making the pith regions. Here there is no central regions freely. All the vascular bundles are occupying the ground tissue, so that here there is no pith. Okay, then differentiation between the, now we saw already dicot and monocot stem. Now we will see the differentiation between the dicot stem and the monocot stem. See, first one, hypodermis. Hypodermis means what? Below the epidermis, just below the epidermis, what layer is present? That is hypodermis. Here in dicot stem, that is made up of collenchymatous cells, unevenly thickened cell wall containing cells. That is a collenchymatous cells. That is present only in the young regions of the plant. But the monocot stem, that is made up of sclerenchymatous cells. Then ground tissue. Here the ground tissue is made up of, in dicot stem, the ground tissue is made up of cortex. Cortex is also upper cortex, middle cortex, inner cortex, and the endodermis, pericycle. With this type of differentiation is present in the dicot stem. But in the monocot stem, there is no differentiation of cortex, endodermis, pericycle and pith. So such a differentiation is not here in the monocot stem. Then vascular bundles. Vascular bundles, all the vascular bundles are present in a round structure of endodermis. In the endodermis, all the vascular bundles are attached, so that is it is a less in number. But here vascular bundles are scattered here and there, so that it is a numerous or many. Then vascular bundles uniform in size. At the periphery to the center, all the vascular bundles are in the round structure, all the vascular bundles are uniform. But here the vascular bundles are smaller in the periphery. Near to the peripheral or near to the border of the stem, the vascular bundles are very smaller. But towards the center region we are going, the vascular bundles are slightly larger. So by going towards the center, the vascular bundles are slightly bigger in size in appearance. Okay, then next one, vascular bundles, all the vascular bundles are arranged in a ring, but the vascular bundles are in monocot stem, it is scattered here and there. And in dicot stem, the vascular bundles are open. Why it is called as a open vascular bundle? Because of between the phloem and xylem, cambium layers are present, so that it is a open vascular bundle. But here cambium is absent, so that it is a closed vascular bundle. Okay, bundle sheath. Bundle sheath is absent. Here surrounding the vascular bundle, sclerenchymatous cells are not surrounding, but here in monocot stem, scleral gymata cells surrounding the overall vascular bundle, so that bundle sheath is present. Okay. Secondary growth. If the gambium is present, the dicot stem is having the secondary growth is also. Secondary growth means side growth or lateral growth, so that the woody plant is forming. But here gambium is absent in monocot stem, so that secondary growth is also absent. Okay. Pith. In the center region, inside the round vascular bundle, large pith regions is present. But here in monocot stem, there is no pith. Then last, medullary rays. Between the two vascular bundles, some parenchymatous cells are present longitudinally, and that is only separated between the two vascular bundles and giving the support also the vascular bundles. So that is a medullary rays that is present in longitudinal in structure. Okay, that is present in dicot stem, but that is also absent in monocot stem. Okay, so these are the very important points of internal structure of dicot and monocot stem. Okay, thank you.